Assalamu alaikum. Welcome dear students. Hello, how are you? I hope all of you are busy in this examination season. ये हमारा एग्जाम सीजन चल रहा है, our pre-boards and the mid-term examination, and बहुत ही जल्दी you all will be appearing in the boards and the final examination in few months. So first of all students, I wish you all the very best in all your future endeavors. Welcome to our YouTube channel, and today our topic of discussion is the transgenic plants with beneficial traits. We have already discussed the various benefits of the genetically modified plants or genetically modified crops, the GM crops. For example, the stress management, stress control, and you know that the stress is of two types, the biotic stress and the abiotic stress. The edible vaccines, the improving nutrients quality, but the certain benefits that I want to discuss in this lecture is the biodegradable plastics. As we know that in this era of technology, the whole of humanity is concerned with how to save our environment, how to conserve our resources, and there is a separate discipline and topic that is known as the sustainable development. That how can we use our resources while consuming our needs so that they can be used, replenished, and they can be used for our future generations. So this is sustainable development. One of the great concerns of the environmentalists is that the non-biodegradable plastics. As you know that in India, in the various developed countries, and in the many third world countries also, there is ban on the non-biodegradable plastics. So students, there is a substance whose name is PHB. The full form is polyhydroxy butyrate. Polyhydroxy butyrate. This is actually a material that is used for making the various type of appliances, the wares, many types of the utensils which are used in the industry, in the research labs, and this substance is of immense use, and this is produced by a biochemical pathway by a particular microorganism, a bacteria that is known as Alkaligenes eutrophus. Alkaligenes eutrophus. So this is a bacterium that produces this polyhydroxybutyrate, and there are three genes which are responsible for the complete biochemical pathway that produces the PHB from its precursor. So as we have discussed in the previous chapter, you must have remembered that in the previous chapter, bacterial cell culture, we have studied about the fermentation technology or the bioreactors. So this in a natural process, because this is a natural process, we can use this particular type of the bacterium for in a bioreactor for the commercial production of PHB. So we can produce the PHB in bulk on a commercial scale using the fermentation technology and using the fermentation of Alkaligenes eutrophus. Kindly make a correction, it's C, not K. Alkaligenes eutrophus. So commercially using fermentation, but the problem is that its production is low, means the yield is low, the problem is, the downside of it, the disadvantage is that it is the low yield, and because of the low yield it is having the high cost. It is having the high cost. So that is the main issue. And you, as you must be well aware of the fact that plants, they are basically the chemical factories, and they use very low cost or no cost nutrients excuse me, the plant cells, they take the carbon dioxide from the environment, they take the sunlight, the radiations from the sun, and they absorb the nutrients from the soil. So, and they produce a large number of primary and secondary metabolites. So we can consider plants as the economical chemical factories. So as we can use the plants as bioreactors, and we can use recombinant DNA technology, and we can insert these 3 genes of which are involved in the biosynthesis of PHB, polyhydroxybutyrate, and we can make the transgenic plants. How do we make a transgenic plant? We have already discussed in this chapter only. In the last few lectures we have already discussed that there are 2 types of approaches, the direct approach and the indirect approach. So using any approach we can make the transgenic plants, and we can insert our genes of interest, which are the 3 genes from alkali genes eutrophus, into the plant cells, and we can make the callus, and we can regenerate the callus into the whole plant, and the plant will produce the PHB in its chloroplast. The globules of the PHB are produced in the chloroplast, whereas the normal physiology of the plant is not affected. The normal physiology of the plant is not affected. So that is basically the beauty. And successfully, this is, I am not telling that this is a virtual thing or this is only theoretically we are saying, rather the transgenic Arabidopsis, you know that Arabidopsis is a model plant species, and we can make the transgenic Arabidopsis which produces the PHB in bulk on a commercial level. Using downstream processing, we can retrieve our PHB, which is the biodegradable plastic, so they are eco-friendly. The best aspect of using this technology is that it is eco-friendly plastic. Eco-friendly. So this is one of the very important use of the transgenic plants. I hope आप लोगों को ये समझ में आ गया होगा. And if you have any doubt, you can ask. You can simply contact me. So on an industrial level, we can use this technology. Now moving to the next topic. So this was one of the very important aspect. Another aspect is the metabolic engineering and the production of secondary metabolites. Metabolic engineering and the production of the secondary products, secondary metabolites. I hope all of you know what are secondary metabolites. This is very important topic. Why? Because number of times it has been asked in the board examination of class 12 that what is the difference between the primary and secondary metabolites. So students, remember that secondary metabolites, they actually exist in both in case of the microbes and both in case of the plants. And as we know that the primary metabolites are the intermediates or the chemical compound, organic compounds which are directly involved in the growth and development of the plants or microbes. Because here we are discussing about the plant tissue culture, so that is why we are more concerned with the secondary metabolite of the plants. And the byproducts of the primary metabolic pathways which are not directly involved in the growth and development of the plant, they are known as the secondary metabolites. In case of microbes, the microorganisms, the example of the secondary metabolites is the antibiotics. And in case of plants, there are many examples like Taxol. There are examples like the sugar alcohol like mannitol. These all are examples, and there is a complete list in your CBSE book of biotechnology. There is a complete list of these secondary metabolites as produced by the plants. So what we do, that how can we produce the secondary metabolites, because they are natural process, but on a commercial scale this natural process, because we need these products of the chemical factories which we call plants in bulk, so that you know that on a commercial scale there is a demand and the supply and demand. So in order to cater supply and demand, so in order to cater to a demand of a large number of population, we have to increase the supply. So what we can do, we can overexpress actually there are two strategies for the commercial production of the plant secondary metabolites. In one strategy, what we can do, that we can, in the first strategy, we can overexpress the genes which are responsible for secondary metabolite production. तो हम इनको ओवर एक्सप्रेस कर सकते हैं. And generally it has been seen that if there is a biochemical pathway like this is a precursor molecule and this is the end product. So the very first enzyme, this is enzyme number 1, enzyme number 2, enzyme number 3, if we overexpress this particular enzyme while making the transgenic plant, so it results in because at every step there is a substrate in the product. For example, S1, S2. So if we overexpress this enzyme, then the more and more S1 will be produced, more and more S2 will be produced, and the product will be increased and enhanced multi-folds. ये काफ़ी बढ़ जाएगा, ये हमें काफ़ी ज़्यादा मिलेगा. तो this is the one strategy that we can use for the overexpression or the overproduction or the large scale production of the secondary metabolites. So it has been this strategy has been successfully applied to the Taxol production. Taxol is a secondary metabolite which is of great importance, great economic importance. So the real example is the Taxol production from the transgenic plants, and Taxol production, transgenic culture of the Texas. It has been isolated from the Texas species. So the transgenic from the transgenic plants successfully it has been implied. So this was the first strategy in which the genes are overexpressed, especially for the first enzyme it is overexpressed in order to enhance the overproduction of our secondary metabolite of interest, for example the Taxol. So this was the real example in which it was isolated from the transgenic Texas species. So this was the first strategy. The second, there is one more strategy, and according to the second strategy we use, you know that there is a strategy that we have already discussed about how to make the plants transgenic. So there is direct gene transfer and there is indirect gene transfer. In the indirect gene transfer, we use a vector-based gene transfer. In case of plant cells, there is a species that we all know. It is Agrobacterium tumefaciens. So there is one more species of Agrobacterium. It is known as Agrobacterium rhizogenes. Rhizogenes. And I forgot to underline the previous name. Always the scientific names have to be underlined. So Agrobacterium rhizogenes we use to induce the excessive formation. What does it do? The excessive formation of secondary roots. Excessive formation of secondary roots. Excessive formation of the secondary roots. And what are the secondary roots? The hairy roots. The hairy roots, they are the secondary roots which come out from the primary roots, the main roots. Okay, suppose this is the main root. So these hair-like structures, the branches, the hair, these are known as the secondary root. And remember that the secondary roots, they are considered as the reservoir and they produce secondary metabolites in bulk. So these are the regions that give rise to the production of secondary roots, the production of secondary metabolites. So we can use the Agrobacterium rhizogenes. We can make this particular bacteriums infect the plant so that it can induce the formation of the secondary roots, and the secondary roots are the sites of the production of the secondary metabolites. So we can overproduce the secondary metabolites, and using the downstream processing and biotechnology, we can then extract our product of interest and we can use it commercially on a commercial scale. So we can use, these were some examples how we can use the plant as a bioreactors. Okay, so you must have appreciated that these are the plants are the natural bioreactors or the natural fermenters. So we can also say that in a nutshell that using these examples, the edible vaccines, the therapeutic proteins, the nutrient quality, and the overproduction of the secondary metabolites, the biodegradable plastics, we can use our plants, or especially the transgenic plants or the GM crops, as the natural bioreactors or natural fermenters. So this we should appreciate. Now, there are some miscellaneous applications also. Miscellaneous applications of the transgenic plants. There are some miscellaneous applications of the transgenic plants. For example, as you know that the oil or the fats, they are in a nutshell, we should know, and most of us know very well, that for the fats and for the proteins, for the best quality of fats and proteins, the sources are different. The proteins we should take, the best quality of proteins are available from the animal source, and the best quality of fats are from the plant sources. And there are some exceptions also. For example, the fish, you must have studied about the cod liver oil. So this is an exception. Otherwise, the healthy oils, they are produced by the plants. But we can using the transgenic recombinant DNA technology in plants, we can produce the healthy oils with altered fatty acyl chains, so we can change the bonds. For example, you must have read about the omega-3 fatty acids, so we can make and we can produce the omega-3 fatty acids using the plants, transgenic plants. And another very important miscellaneous application is that we can modify our starch properties, because starch is the food reserve for the plants. So the modification of starch properties, it is another very important application of the natural bioreactors like plants, and we can use our starch in different ways. Then a third very important application of the transgenic plants is, it is very important, the favorable change of the grain storage production. Favorable change in grain storage products. What does it mean? It means, what is a grain? It is a seed, it is a cotyledon. In the cotyledon, the two main components are the embryo and the endosperm. Endosperm is the main reservoir of the starch. So using the various genes and the promoters which are involved in the endosperm, we can do we can make our own product of interest in the endosperm of the starch grains. This is another very important, for example, we can produce some specific proteins containing the essential amino acids, and the chemical composition can be changed. And for example, if we take wheat and we have made, suppose the transgenic wheat, and we can induce some proteins, we can express or produce some proteins out of it, then we can use this wheat flour for the bread making. So the bread which will be from this type of the wheat will be rich in proteins. So that is one very good example. And for malting of barley and the brewing of beer, means the hard drinks, the beer and the other things also, because malt and very important, the barley and the brewing of beer, the malt is used. So we can use these grains for making changes in the composition of beer and the barley products. So these are some examples that we should take into consideration. The fourth very important example, miscellaneous example, is the removal of the undesirable toxic compound, removal of toxicity. As you may be knowing that the certain plant products, they are toxic, they have certain side effects. They are good for us, but they have certain side effects. Okay, so the side effects or the toxic effects which are detrimental for our health or for the normal physiology of the cells, these have to be removed. So using the transgenic plants, we can easily remove the undesirable toxic traits. And then aesthetically, the fifth one is also very important, and we can make the desired characteristics in the flowers. So aesthetically, you know that what is the significance of the flowers. When some dignitary comes to our school or college or university, we present the bouquet, which consists of the various types of the flowers. The colorful flowers, they are very soothing as aesthetically. So there are certain colors and the pigments that actually exist in nature. For example, we know about the red roses, pink roses, and the yellow and the white roses. But the blue roses, they are not natural. So we can produce the different colors which are not natural, different colors of the flowers which are not natural. For example, the blue rose. The blue rose in your book, CBSE Biotechnology book, on the page number 130, there is a diagram of the blue rose. There are two different shades of the blue rose, and the pigment that confers the blue color, their gene has been incorporated using the recombinant DNA technology, and because the blue pigment is not very common, and the blue and certain other types, certain other colors can also be produced. And same is the case of another very good example is the colored cotton. You know the cotton wool is white, but we can make the colorful or the colored cotton. So using the same technology, the colored cotton can be produced, and which is not natural, you know, but using the transgenic technology, the recombinant technology, we can use the colorful cotton. Then another very good example is the production of the tearless onions and the caffeine-free coffee and the low nicotine tobacco. So we can use our products of interest, and we can remove the toxicity, and we can make our products in the desired form. For example, I just gave you the example that the tearless onions, so that you can eat, you can use, you can cut, and you can grind these onions without having any adverse effect on our eyes and normal physiology. And you know that coffee is a good source of caffeine. Caffeine is a relaxant, but after a particular threshold or limit, it is not very good for our health. So using this technology, we can make the caffeine-free coffee. And you know that tobacco in the third world countries, especially India, it is one of the causes of the cancer because it is very rich in nicotine. So we can make the low nicotine tobacco. So we can kindly make a small correction. It's NICOTINE, low nicotine tobacco. So there are many important and exciting applications of the transgenic plants. In a nutshell, we can say that they are the natural factories and the natural fermenters and the bioreactors which can be exploited using recombinant DNA technology for the production of our products of interest, proteins of interest, the stress management and the removal of toxicity, the removal of certain characters which we do not require. And we have seen the complete list of the various type of the applications. I hope that you must have enjoyed, you must have understood, and आपको ये समझ में आए होंगे ये सारे topics. They are very simple, straightforward, and when you will go through the book, you can further understand these topics in great detail. and आपको जो भी problem हो, if you encounter any problem in the understanding, kindly contact me personally on my WhatsApp, on email, and then we can be in touch and the things will be more clear. So the last topic of this chapter is the biosafety in the plant genetic engineering, that we will discuss in the next online lecture. Till then, take care, study well. This is the exam season, pre-board season, and this is a midterm season. So I wish you all the very best of luck, students, and bye. Take care. Allah Hafiz.